So you're here to learn about cells, huh? Well, let me tell you, the life of a cell is not easy. A cell goes through two phases of the cycle, interphase and M-phase. I'm going to go through interphase. Watch and learn. So you can see that I'm a lot smaller than I was before, which is really saying something considering how small cells are. So the first thing we have to do is change that. And now I have to double all of my organelles for my new cell. And now we're ready to start synthesis. These chromosomes are tightly packed strands of DNA. For DNA replication, the first thing that needs to happen is an enzyme called helicase needs to break the hydrogen bond connecting the double helix together. Then an enzyme called primase comes along and puts a little marker called a primer down, marking where a new strand of DNA needs to be constructed. Then an enzyme called DNA polymerase comes and builds a new helix mirroring the first helix. This will happen a few times in a few different places along the double helix. If there are gaps in the helix, an enzyme called ligase will come along and glue the gaps together. The end result is that each chromatid is replicated into an identical sister chromatid.
So now I need to grow a little bit bigger. Hold on a second.
That is much better. Now I just need to create a few structures called centrioles that will reach spindle fibers out to my chromatids and pull them apart. And now we're ready to start mitosis. I'll take my time a little bit more this time. The first step is prophase, where all of the chromatids compress themselves a lot smaller, and also the nucleus starts to dissolve. Metaphase is when all the sister chromatids line up in the middle of the cell. This is when the spindle fibers reach out and attach themselves to the chromatids. Then there's anaphase, when the chromatids are pulled apart to each side of the cell by the centrioles. Last up is telophase, when new nuclei are forming and the cell is ready to divide. And then there's cytokinesis, which is basically the splitting of the cell into two new cells. So there you have it. Those are the stages of a cell life. It's pretty complicated, right? Well, luckily for you, this part of the tour is over. Not so fast! There's a whole other part of the cell cycle that needs to be considered. If you want to be an egg or a sperm cell, a gamete, then you need to do meiosis. Meiosis is a lot like mitosis. However, with meiosis, you need to do PMAT, prophase, metaphase, anaphase, and telophase twice. With prophase 1, along with all the chromatids getting a lot more compact, they also find their homologous pair. A homologous pair is two chromosomes that contain the same type of genetic code in the same area, but one chromosome came from each parent at the child's birth. In this phase, there is also a possibility that the two chromosomes will cross over and exchange their gene code with one another. Then comes metaphase, where the homologous pairs line up in the middle, and the spindle fibers from the centrioles attach to one chromosome from each homologous pair. It is completely random as to which chromosome the spindle fiber will attach to. Then comes anaphase 1, where the chromosomes are being pulled to each side of the cell. And finally, telophase 1, which is where the nuclei are forming around the new chromosomes. And cytokinesis 1, where the cytoplasm of the cell is splitting into two new cells. Then comes prophase, metaphase, and anaphase, and telophase 2, which is basically the same as mitosis, except with half the number of chromosomes. The end result is 4 egg or sperm cells, 4 gametes. So there you have mitosis and meiosis. And now, let's move on to budding. Wait, what? Oh, come on, why did I have to be the bud? Oh, stop complaining, you look like number 1. I feel like number 2. Budding is when a new cell grows off the first cell. It then pops off and grows off into a new cell. Many organelles are injected into the bud. Wait, what? Ugh! And then the bud separates from the main cell and grows bigger into a cell genetically identical to the original. Are we done here? Oh, we are just getting started. Aw, crap. Asexual reproduction versus sexual reproduction. Asexual reproduction requires one parent and produces 2 genetically identical children. Sexual reproduction requires 2 parents and produces 1 child with traits from each parent. Asexual reproduction requires a lot less time and also doesn't require a partner. However, the exact replication provides no variety in the gene pool. Sexual reproduction requires a lot more time and also, of course, requires a partner. But, however, this can lead to a very diverse, strong genetic gene pool. Hey, do you want to watch this show on Manx syndrome with me? I have a nice comfy bed for us to sit on. No. What is Manx syndrome? Manx syndrome occurs most often in a specific type of feline, the Manx cat. The Manx syndrome causes the spinal cord and, particularly, the tail to grow abnormally, and sometimes leads to no tail being developed at all. This can impact the cat's movement ability in varying degrees of severity, from a slight hobble to needing to hop around everywhere to be able to move. In addition, this genetic disorder leads to disease of the nervous system, incomplete forming of the vertebrae, and involuntary urination and defecation. How is Manx syndrome manifested? Manx syndrome occurs when a cat has a mutated M chromosome. The M chromosome probably would have mutated during meiosis, during DNA replication. Just one mutated M chromosome is required. 2 would be lethal, and usually leads to an early death of the kitten before birth. Since it is a chromosomal disorder, it can be passed down to kittens. If one parent has the genetic disorder, there is a 50% chance that the kitten will have the genetic disorder as well. If both parents have the genetic disorder, there is still a 50% chance that the kitten will get the disorder as well, but this also introduces a 25% chance that the kitten will receive both mutant M chromosomes and die before birth. What can be done about this disorder? Not a lot really can be done at this stage of our technology, unfortunately. If in the future we could figure out what causes the mutant gene to express itself, we might be able to reverse, or at least dull, its effect. As of right now, continued breeding of Manx cats with Manx syndrome will cause the problem to continue, greatly impacting the cat's movement ability, general quality of life, and even ability to communicate with other cats, since tails are a primary method of communication and is therefore simply unethical.